Hello, welcome to this video. This is a crash course in molecular biology, originally prepared for the 2019 iGEM Spring Course and now recorded for the benefit of the 2020 team. In this video, I'll give a review of what's known as the central dogma of molecular biology, and I'll also highlight one of the classic examples of how the expression of genes can be regulated, the example of the Lac Operon found in some bacteria. The central dogma of molecular biology, the main point behind the genetic information you find in all living cells, is that these genes mostly function as recipes for how to build proteins. Proteins are complex molecules, chains of amino acids, that are responsible for many of the structures in the cell and the activities. By linking together certain amino acids in a specific sequence, we'll cause a protein to adapt a certain shape. And the shape of the protein defines its function. The idea of structure and function being linked should be a somewhat familiar one. If you look at a set of tools, each tool has a different structure and is used for a different function. You wouldn't use a screwdriver to act as a hammer or vice versa. The genetic information that you find in the form of DNA, sometimes packaged in molecules known as chromosomes, are the inherited copies of many different genes for making many different types of proteins. The DNA form the permanent records, the cookbook, if you will, containing all the recipes every one of your cells will need to make any protein found throughout your body. Just as a chef is not simultaneously cooking every dish in a cookbook, in the cell, the DNA recipes, or genes, will first be transcribed or copied into a similar chemical known as RNA. Whatever the working recipes or the proteins that are needed at the moment will be expressed first through this transcription step. Then, specialized machinery or molecules in the cell, for example, one of the central ones here is known as the ribosome, will then take the genetic information and read it. And this information again specifies the order of amino acids to form a particular protein. That protein is the finished product and can carry out some activity. If you want to create a behavior in the cell, you're going to need to find a particular gene or recipe for making a certain specific protein. The amount of that protein you make depends on the control you place over the process of transcription and translation. In most synthetic biology projects, and in many natural cellular examples, the main area of control is on this process, transcription, in which an enzyme known as RNA polymerase, together with other proteins, has to find a particular recipe and produce it at a certain rate, or transcribe it into RNA and express it at a certain rate. How is the beginning of a gene identified? This is normally done through a part of this gene known as the promoter sequence. This is information contained just upstream of the coding region of the gene. This is the area in which RNA polymerase will bind to begin transcription. If we want to continue this analogy of a recipe, while a recipe will contain specific instructions for ingredients, you normally will have a chapter title or recipe title, which gives some context and direction for when you should produce this recipe. This is what a promoter is for a gene in DNA. Certain promoter sequences, although they are not recognizable to us, will be recognized with varying degrees of transcription by RNA polymerase. So there are ways to control whether or not a promoter leads to a high level of transcription or a low level of transcription. Here is a depiction of how in bacteria such as E. coli, RNA polymerase actually contacts the DNA, reading these certain sequences and determining whether or not transcription should begin. As you can see, the protein here needs to physically interact with the DNA to read this sequence and determine if there's a promoter. So even if we have a promoter present, if we somehow blocked this interaction, we would limit transcription. If we otherwise encourage these two molecules to interact, we would promote transcription. So there's a level of control that can be placed. Transcription can be dynamic. This also can be set up so that transcription controls more than one gene. You can have one promoter create the RNA and the recipe for making several different proteins at the same time. So we can start to imagine ways in which different genes and promoters might be combined to create a sort of circuit which will control a range of different behaviors at the same time. Here is an example of such an arrangement. This is known as an operon. Here we have a promoter element to the left. Our RNA polymerase can bind. If this binding is promoted, we'll create a molecule of RNA, here shown as a sort of salmon-colored ribbon, and this within it will contain the recipes for several different proteins. The reason why I'm highlighting this particular arrangement of genes is because this is the arrangement for a classic example of how the expression of genes is regulated, and this example is the lactose metabolism operon from bacteria, usually just referred to as the Lac Operon. Here is a rather busy slide showing the lactose operon, or the Lac Operon. We have here, part of this looks like it did from the previous slide, where we have RNA polymerase bound to a promoter. Following this are a number of genes which the cell will want to activate when lactose is present. These are proteins which will act to speed up degradation of lactose. We also have, to the left of this image, another protein that's being produced under the control of a different promoter. And this protein doesn't have a function in the breakdown of lactose. Instead, this protein, its shape, its function, serves to interact with that promoter and potentially block RNA polymerase. This is a repressor protein. So, again, when thinking about how this system is going to be regulated, think about why do you want to express certain genes at certain times. Expression of a gene costs time, raw materials, and energy for a cell. So if there's not a need for this gene, you're not going to want to spend the time to make the RNA and make the protein. However, if lactose is the only source of energy for this cell, it's going to want to turn these genes on. Thinking about this will allow you to make sense of why this operon is regulated in the following way. There is a protein, the one we mentioned a moment ago from the left-hand side of the diagram, known as a repressor. It's called the Lacl transcription repressor. Normally, when this protein is made, it will bind to a sequence which overlaps or is right next to the promoter. If you remember before, we said that for RNA polymerase to express a gene, it needs to physically be able to interact with the DNA. This protein binds the same sequence and will block that interaction. So as long as this Lacl protein is around and is in an active shape, it will actually block transcription of the Lac Operon. However, if certain molecules are present, such as lactose or a chemical analog known as IPTG, this will bind to the Lacl protein and cause it to change shape. Once it changes shape, it's inactive and no longer able to repress transcription. This is the way that repressors and promoters can work together to control the expression of an operon or a gene. This brings to mind the saying, the old adage, the enemy of my enemy is my friend. So here, lactose or IPTG or allolactose is the enemy or the inactivator of the repressor, which was the thing repressing the rest of the Lac Operon. So this allows you to begin expressing these genes. For that reason, those small molecules like IPTG are referred to as inducers. They lead to induction of this gene. Here's a quick way of visualizing what will happen. If you add lactose or IPTG to cells with this operon, they will begin to create RNA copies of that gene and begin to translate it into proteins. So you will see expression of those genes increase over time. Now, for many genes, this is not an absolute process. So even without the inducer, there might be some small background level of expression. This doesn't tell the full story of the Lac Operon, however. There are ways in which you can layer on top of a promoter more than one type of signal. So here's another example from this same set of genes. In addition to that Lac-I repressor, there's another protein known as the CAP Activator. This will respond to low levels of other sugars in the cell, such as glucose. So this is directly responding to levels of a molecule known as cyclic AMP, and these are an indicator of glucose starvation or low energy in the cell. So the presence of this CAP activator means that the cell is starved for energy and there's not a lot of other sugars around. So again, thinking about how this should impact the Lac Operon expression, we can realize that when you really need a lot more energy, you're going to want to maximize that lactose metabolism, because apparently metabolism of other sugars is not enough. Right? Again, it's not enough because there's not enough energy in the cell. So if we took a closer look at that promoter, we'll see that actually there's another portion of this sequence, which is where this CAP protein will bind. Again, a main thing I want you to think about for this, in terms of synthetic biology, is that this promoter region is a sequence that's going to be defined by different features. It's going to have some features which allow it to bind to act as a site where CAP will bind, some features which will allow RNA polymerase to recognize it, and some features or parts of the sequence which will allow Lac-I to bind. When looking at different parts from an iGEM collection, different BioBricks, often these sequence elements will be defined and characterized, and you can mix and match and combine them together as needed. Here again is showing the response that we get from this CAP binding protein. When there is plenty of energy in the cell, CAP will be present but inactive. It won't be able to bind DNA. When there is low energy in the cell, we'll have cyclic AMP. This will activate CAP. CAP will bind then to the DNA and actually help to stabilize and recruit RNA polymerase rather than blocking it, which is what we saw the repressor do. By layering different types of DNA binding proteins on top of each other, we're able to get a more sophisticated type of response. Here we have not only high expression and low expression of the gene, but intermediate levels depending on the exact amounts of lactose and glucose. So, by way of review, we can control the expression of a gene through the region or the part of the sequence of that gene known as the promoter. Different promoters promote different levels of transcription, and these can also be influenced by the presence of different repressors or activators. There are a wide range of different repressors and activators and different promoter sequences which can be used in different genetic circuits. There are other ways of thinking about this. We can actually abstract the relationship between these different molecules. This is a useful thing to do Instead of worrying about the actual structure, the DNA sequences, just think about what kind of behavior do you want the cell to produce? How are you going to connect these? This kind of interfaces with the types of ways you might predict the behavior of this system. You can actually do some math modeling of a system by abstracting how these genes and sequences are interacting. Here in this depiction, you can see that Lacl is repressing the expression of the Lac Operon, Lac Z, Y, and A. IPTG will actually stop the repression, and expression of these genes actually destroys things like allolactose or IPTG, in theory. So the way we can predict how this circuit would behave would be a negative feedback pattern. You add the inducer, if it's, let's say in the case of lactose, you add it, you'll turn the genes on, but eventually the inducer will be broken down through the products of those very genes, and the cell will return to the initial state. You can see some of these same elements that we discussed when looking at an actual BioBrick. So here's a very commonly used BioBrick, BBAG04450. This is a RFP, or red fluorescence expression BioBrick. And we can see actually some of these same elements that are going to be important. So we have a promoter sequence, a region of the DNA which is responsible for activating transcription and allowing that gene to be turned on. If you want to produce some protein, some function in the cell, you're going to need to have a promoter somewhere in your BioBrick or in your design. In this case, we actually have the very same promoter we've been talking about, the Lacl promoter, which in the collection is known as R0010. This will allow transcription to initiate, will create an RNA. That's not the only signal we'll need. I hadn't discussed this in the video yet to great detail, but you also need a signal to initiate translation. This is known as a ribosome binding site. Here's a very commonly used one, B0034. Notice that the relative positioning of these is important. You can't have the ribosome binding site before or upstream of the promoter, or else it wouldn't be present in the RNA that gets created. This then is followed by the coding region, the instructions for making a specific protein, in this case a protein which is endowed with the ability to fluoresce red under certain conditions. And finally, there are some stop signals to tell RNA polymerase that it's finished transcribing a gene and it doesn't need to continue. You don't want the genes, let's say, next to this or downstream of this particular circuit or block to be activated. This is a way of insulating this from other parts of your genetic circuit. Thanks for watching.